Okay, so the first step in this is to get our sample, and I'm going to go for blue spruce. So here's blue spruce right here. I'm going to cut a nice little sprig off of blue spruce. The next step is to get the mass of the sprig. To get the mass of the sprig, I'm going to take a little bit of weight paper, place that weight paper on the balance, tear the balance so it zeroes out, take the sprig, put it on the balance, and you can record that mass of the sprig. The next step is to set everything up and get it all ready for photosynthesis. Here is our CO2 gas probe. All right, so this is going to measure the amount of CO2 that's in the chamber. We have then our chamber here for that we're going to place our sprig in. So I'm going to take the sprig, place it in the chamber, make sure that it's all spread out inside that chamber there. Okay, move it like that. Then we're going to place a heat sink on top, because when you put light on top, light has heat. I'm going to put this in. This CO2 probe is hooked up to a computer. The computer has software running. The software is called Logger Pro, and it's from Vernier Scientific. I'm going to go ahead and get all the aluminum foil around it so that we maximize the light going in. Finally, then I'm going to add a couple of lights. So here is a light source. We'll put this light down over the top of it and get it going. And I might as well even make it super high light intensity. We'll get 2 lights on top of that guy and turn them on and get them going. Okay. All right, here's the computer program all set up and ready to go. So we have our computer on here. Our program's called Logger Pro. Over here we have our CO2 live reading from our CO2. Our y-axis is CO2. We have minutes here. We're going to run the experiment for approximately 10 minutes. So the first step is basically to take my mouse. I'm going to go up here to the top. I'm going to click play and get this thing going. So there we go. And it's starting to record or take readings. If this is photosynthesis, we have our plant underneath the light. Then it's going to start this line right here, and you can see this line is going downward. I'm going to adjust the y-axis a little bit because it's all the way up here at 1000. So I'm going to take my cursor and change that to more like 800 so that we can see that a little bit better. Okay, looks good. Then I'll show what happens after 10 minutes. Okay, since I gave this a lot of time to equilibrate, I think I'm just going to run each of these trials 5 minutes. We have a nice steady downward curve, slightly slowing down right down here, but good steady curve. We'll shut it off right at 5 minutes and then get our next treatment going. Okay, that was our first treatment. It's completed now. So we have to set up for our second treatment. I'm going to turn these lights off, move the lights out of the way. We'll take the CO2 gas probe out, and then we will basically air out the chamber. So we have to get all that air back out there. So we want to return inside that chamber to regular ambient air. Okay. After we air it out for a little while, then we're going to reattach the probe. I'm going to turn on a light, though this is going to be quite a lot farther away. Alright, I've given it a few minutes to equilibrate, get all of the photosynthesis and everything working properly. I'm going to go up here and start this, and we're going to run our second trial and see what's happening. Now, by airing it out, we want it to start at almost the same level because we don't want the CO2 to be a factor in here. And if we can always start it with the same amount of CO2, then that would be more consistent. Here's our line going. We're going to run this then again for 5 minutes. I might adjust that y-axis now because it looks like it's a teeny bit higher. It might be going up, so I'll click on that 800, and let's change that up to 1,000 so that we can see that line, we can see what's happening. Okay. And this one here is actually going up a little bit, but I'll turn it off and then we'll look at it again at the end. Okay, now we're almost at the end of 5 minutes. We'll turn it off at the end of 5 minutes. But this line is slightly going up, fairly consistent trend, slightly going upwards. So somehow a little bit of CO2 is getting into that chamber. And we're not just getting a downward slope. So we had a downward slope at first and a slightly upward slope here. We'll shut it off right there at about 5 minutes. Okay, and stop. Okay, for our third treatment, I'm just going to put the lights a little bit closer. So since our CO2, we'll just actually make it, we'll just put on 2 lights. A little bit brighter, so we'll just kind of double that intensity there, but have it still pretty far away. So it's not going to be as intense as the first one, but it'll be more intense than the one we just did. Since our CO2 was at a pretty good level, then I'm not going to worry too much about airing it out. Okay, now I'm going to start the third treatment, right, with the lights kind of midway between both of the last ones. Start it here, and you can start watching that line begin. Again, the CO2 is right about in the middle. So we're doing well there with the CO2. And then I'll let it run again for 5 minutes. We'll look what happens after 5 minutes. Okay, close to 5 minutes, or close to stopping this. So we have this red line being the most light, green line being 2 lights, fairly close, a little bit less light there. Blue light actually goes up, which is even less light, but it did have light on it. All right. And so that's where we are right now, and then you have to think about what's going on and how you're actually going to measure photosynthesis with this information. So I'm now going to go up and hit stop and prepare then for our final trial. Okay, for our final trial then I'm going to turn off both of these lights. We'll remove this. We're not going to worry about the CO2 because you'll find out in a minute. And we're actually going to put it totally in the dark. We'll see what happens when it is completely in the dark. Okay, now we're ready for our final piece of data collection. And so I'm just going to go up here and start it. Right. And so we had our 3 lines here, and then we should have our 4th. I didn't really air it out, so our 4th is starting a little bit lower. But we'll see what direction it goes and see if that's a problem by the end. All right, so I'm going to let it run another 5 minutes. Then at the end of the 5 minutes, we'll stop it and we'll prepare for the data analysis. The next step is to figure out what's going on with these lines and actually gather the data that you'll be using in your analysis. Since we have pretty much straight lines, this is a slight curve, a slight curve, a slight curve, not fully straight, but they're straight enough. We're going to use a linear function to estimate the rate of photosynthesis or whatever other processes are going on. So to do that then, I'm going to highlight. Oops, let me get this better. I'm going to highlight from the start here over, and we'll highlight for the full 5 minutes. Up here then you can see this line tool, so that's a best fit line. I'm going to click on that. Then it asks me for each run, so I'm going to select all 4 runs. And we'll click there, and then we have our data for all 4 runs here. Okay, so we can move those over and kind of see which runs they belong to. So that's for our last run. That's actually for our second run. That'll be for our third run, so that's good. So that run, that run, that run, that run. Now we'll go ahead and just focus in. so that you can see those values clearly. Okay. And think about the line. So if we're talking about the best fit line, we're looking at rate. The rate is going to be slope. Since we have y equals mx plus b, m is the slope, and so you'll be recording the slopes of these lines in parts per million per minute. Ultimately, you will divide by the grams that we started with, so that it's parts per million CO2 per minute per gram. Next, you should go to the lab report and fill out question number 2. Question number 2 asks you to determine the rate of photosynthesis at the 3 different light intensities using these data.